Okay, good afternoon, everybody. I hope you can all hear me. Can you hear me? Just somebody say yes or no.
Yes.
Yes.
Okay, fine. All right. So this afternoon, we're going to move on a little bit because up till now, what we've talked about the molecules that make up living things. We've talked about how those molecules are organized. We've talked about some of the aspects of metabolism, about how matter and energy interact in the physiological processes of the organisms. Now we need to start talking about the organism, the entire organisms themselves in their environment. And the way we're going to start off doing that is by discussing how it is, in actual fact, that microorganisms grow, especially prokaryotes. From now on, most of our discussion is going to be about prokaryotes and not about the eukaryotes. So we've spent a little bit of time talking about microbial growth in one way and some of its implications, especially in health sciences, since so many of you are involved in health sciences. So of all of the processes that we've talked about, these things constrain organisms, that they have requirements that they need in order to grow, in order to survive in their environment. The first set of constraints are those of the physical environment itself. Remember always that when we talk about the organisms, we're talking about those complex interactions of metabolic process. And especially we're talking about the functioning of those huge molecules, the biomolecules, such as the enzymes. We saw that those molecules operate under strict conditions. They have conditions where they operate best, conditions where they operate really poorly, even conditions where the molecule is effectively killed by its environment if the environmental conditions become so severe. So that places a set of physical requirements on the organism so that all of those functions can function optimally. They require an optimal temperature, largely determined by their enzymes, by the way, which have an optimal temperature. They have an optimal pH. Many of the metabolic processes that we talked about, aerobic respiration, all of those things, they are processes where the organism is taking ions and moving them around, especially protons, for example. If you start changing the pH, you start affecting that tremendously. You also affect the functioning of the enzymes. So there's an optimal pH. There's also an optimal osmotic pressure. The osmotic pressure from the environment relative to the cytoplasm has to be exactly right for what the organism is adapted to. In most cases, the osmotic pressure needs to be, the exterior osmotic pressure needs to be a little less, at least, than the osmotic pressure inside the cytoplasm so that the cell continually expands. But there are some special cases where osmotic pressure outside can be very high. They can be specially adapted to it. Organisms also have a set of chemical requirements. We understand, first of all, that the organisms need things like carbon, nitrogen, sulfur, phosphorus, all of these sort of things. We've talked about those for example, in the construction of the biomolecules. So you understand why. There may be some that you have not heard about. For example, they need very small amounts, sometimes of metals, such as iron, molybdenum, all of these sorts of things, which often are involved again in enzyme function. Depending on the organism, many organisms require oxygen. They may also require from the outside other growth factors, such as vitamins, this sort of thing. It depends a lot on which organism you're talking about, because organisms are highly adapted for their environment. And especially when we talk about prokaryotes, there are many different kinds of prokaryotes, and they are adapted to many, many different environments. Some of them extreme environments, extremely hot, extremely acid. In health sciences, most of what we talk about when we talk about human beings, where the microorganisms are adapted to the body temperature of their host, to the osmolarity of the surrounding fluids of the host, etc. So when we talk about human-associated prokaryotes, we would usually expect them to be operating around about 37 degrees Celsius, our body temperature, and around about our pH, which is just a little bit less usually than the neutral, and at our osmotic pressure. That is the saltiness, solute concentration of our blood and our body fluids. We'll look at it in more detail. So here, for example, are the growth rates of different kinds of microorganisms in response to temperature. And we actually classify prokaryotes or organisms in general in terms of the optimal temperatures that they grow at. So have a look here on this axis here. This is the rate of growth. So let's just take the very first one. Rate of growth is very low here. It's high here, and it's again very low here. So this is the temperature axis. So let's just have a look at this very first one. It has an optimum growth temperature, which is around about there, about 11 or 12 degrees Celsius. That's where its growth rate is the highest. But that's not to say that it doesn't grow at anything but that temperature. It just grows best at that temperature. And the reason for that is that all of its enzymes, all of its metabolic pathways are adapted to that particular optimum. You'll remember we said about enzymes, they have an optimum. And then they have a maximum and a minimum beyond which they don't function at all. That puts constraints on the growth that we see here. This organism put together into an organism, all of those biomolecules now are functioning at an optimum temperature. There is a minimum temperature below which they simply won't grow. That is not to say that they die. At this low temperature here, they may simply stop functioning altogether, but not die. But the same is not true about this maximum temperature here. When the organisms reach this maximum temperature, very often they are killed because the enzymes themselves are denatured. They simply lose their structure and are unable to function anymore. So they have a maximum and a minimum. Between that, they have varying rates of growth. But take them away from this optimum temperature and the growth rate falls. So this group of organisms, look at what they are constrained to. They are constrained to an environment between about -8 degrees Celsius and about 18 degrees Celsius. And those are their extremes. There's hardly any growth there. Their optimum is around about here, about 12 degrees Celsius, which is cold. These are organisms, there are many of them, which are adapted to cold water. In places, for example, you might find them environmentally, you might find them at the poles or in seawater, deep seawater. In cold environments, they are adapted to that and they don't function well outside of it. So those are, we call it psychrophiles. And they have an obligation to grow in a cold environment. The next group here, if you look at their optimum, it's around about 22 degrees Celsius, which is approaching room temperature. Again, they have a maximum of around 30 degrees Celsius and a minimum at about 0. So they are constrained between 0 and 30 with an optimum here around about 22. So they're growing in cool environments. So this would be outside of a mammal, etc., in a cool environment, in the soil maybe, or something like that. And those we refer to as psychrotrophs. They prefer cool environments, but they can actually grow around about normal environmental temperature as well. The next group are of particular interest to us, because look where their optimum is. Their optimum is here at around about 37 degrees Celsius, 35 to 37 degrees Celsius. So that is the temperature of most mammals. And those, we could expect that these would be prokaryotes which would be associated with us. So their optimum is around about 37. Their minimum around about 10 degrees Celsius, and their maximum right up here around 48. So they actually have quite a wide range of temperatures, but their preferred temperature is around about 37. That is their optimum. These we call them mesophiles. And mesophiles encompass most of the prokaryotes which are associated with us. Also many others as well. There are free living ones that have the same kind of limits, but those associated with us, not only pathogens, but all of our healthy bacteria, all the bacteria which are normally associated with us as well, are around about here. And those we refer to as the mesophiles. The next two groups are extremely interesting. The first is, look at this here, this group here. They have a optimum around about 60 degrees Celsius. You would find it, if I gave you a vessel with water heated to 60 degrees Celsius, you would find it difficult to hold on to it. Your hands would be burning and you'd put it down. This is very, very warm. And this group have enzymes and everything which are especially adapted to that higher temperature. And look at their maximum around about 72 degrees Celsius, at which point you would actually be burning, if you would scald yourself if you spilled that water of 72 degrees Celsius on yourself. Their minimum is 40 degrees Celsius, which is really warm, really, really warm. Well, that's 3 degrees above our body temperature. That's their minimum. And their optimum around here, around about between 60, 62. This is a group which are specially adapted to growth in hot water. And the places you might find them are areas where hot water occurs naturally, such as hot springs, those sorts of environments. And they used to be thought, first of all, their discovery was monumental. People were just amazed that there were things that grew at these high temperatures. Some of them have proved to be extremely useful to us industrially and in genetic engineering, all sorts of different things, which I'll tell you about later on. But these are called the thermophiles, those that love high temperature. They're called the thermophiles. And some of the most famous have been extracted, for example, from hot water springs in Yellowstone National Park, those kind of areas. It used to be thought that they were extraordinary and exceptional, and that there could not possibly be anything that would actively grow and metabolize and everything at a higher temperature. But then it was discovered that there is this group here. Look at their optimum. Their optimum is around about 95 degrees Celsius. That is close to boiling, and they will, in fact, continue to boil. Here's 100 degrees Celsius. That's the boiling point of water at sea level. So these organisms are growing quite happily. In fact, they require a very high temperature, and their optimum is between 90, 95, 97, around about there. Their minimum 65. Let me tell you something. The process of pasteurization, we heard about it before. It's a method of not sterilizing, but of largely clearing a liquid of actively growing bacteria. It was a way of eliminating pathogens and spoilage organisms from milk, for example. When you pasteurize something, you raise it to 65 degrees Celsius minimum, and you hold it there for about 20 minutes. Then you cool it and you repeat it usually. 65 degrees Celsius is usually sufficient to kill almost all bacteria. But here we have the hyperthermophiles. That is what their name is, those that love extreme heat. And these, well, the first thing you have to ask yourself is, where on earth do we find boiling water? Well, you might find it in hot springs and things, but you find water over 100 degrees Celsius only in areas which are pressurized. So these hyperthermophiles are typical of areas, for example, at the bottom of the ocean, volcanic vents, at hot water vents on the floor of the ocean where they're under tremendous pressure and the water does not actively boil. Nonetheless, they can survive at these extraordinarily high temperatures, and they have highly adapted proteins to make up their enzymes and everything, and they have highly adapted nucleic acids, which are closely associated with proteins that protect them from the effects of boiling. Everything else, you raise the temperature of nucleic acids above about 75 degrees Celsius, the nucleic acids actually melt. But in these that are adapted to high temperatures, all of their macromolecules have special protective mechanisms that have evolved. So know these groups, and at least know their optima for the different psychrophiles, psychotrophs, mesophiles, thermophiles, and hyperthermophiles. And we are also going to, when we talk about environmental bacteria, there are some down here which we're interested in terms of human health, because there are a few pathogens which will continue to grow down here in these lower temperatures. And those become interesting to us, especially in terms of food science, things like that. I'll show you in a while. Here you can see these are things which, these are organisms which we are interested in, especially in terms of their causing food spoilage, number one, but especially causing disease transmitted by food and liquids. So it's food poisonings of one sort or another. And in modern times, we have to deal with this far less than our ancestors did, because we can refrigerate things. We can actually take foodstuffs and we can put them into a fridge and keep the temperature really low. And we hope that things like all the mesophiles will practically stop growing. Down here, around about here, is where our normal fridge sits. And down here is where the freezer sits. So we hope that most of our pathogens and things that can cause us disease will simply stop growing at these low temperatures. They grow very, very well if the food is kept warm and is not refrigerated. That we understand. So first of all, here's the real danger zone for food which has been contaminated with either spoilage organism, which is going to cause the food to rot and not be edible, or things like salmonella, those kind of food poisoning organisms. They grow extremely well in this region here. So here's 37 degrees. So that's our body temperature. Many of them adapted to that as their optimum, but they will continue to grow at these temperatures. This we regard as being a danger zone. We try to avoid keeping food and in this danger zone for very long if it is subject to spoilage. If we drop the temperature below that, for many of them, we will stop them from growing well, but not totally stop the growth. If the food is kept at those temperatures for long enough, the bacteria can still accumulate, and they can really importantly, they can continue to produce any toxins and everything that are inherent to them that might cause us illness. So many of them will survive, continue to survive. And there's one particular food poisoning organism which causes tremendous problems called Listeria, which grows quite well at these low temperatures. But if we get lower than that, very, very few bacteria will grow close to, very few bacteria of concern will grow close to freezing. And below freezing, there's really very little significant growth. On the other end of the scale, most pathogens and most spoilage organisms grow very slowly between 50 degrees Celsius and 60 degrees Celsius. And above that, actively growing cells are actively killed, particularly if they're held in this temperature range for any length of time. That's the principle of pasteurization. We take things and we heat it up just hot enough that it won't ruin the food stuff, but at a temperature somewhere here which is disadvantageous to the pathogens or the spoilage organism, and we kill them. It works really well, except for one thing, and that is, remember back that I told you that there are groups of gram-positive organisms, the Clostridia and Bacillus, which are able to produce spores. And those spores are completely resistant in this range here, up to, in this range, right up to about 110 degrees Celsius. They are completely resistant. And if you want to kill off bacterial spores, you need to bring them up to this range of temperatures here. And in order to get water above 100 degrees Celsius, we need to pressurize it. So this is the principle of autoclaving. An autoclave is basically a pressure cooker. It's a system where we allow steam to be produced to such an extent that it pressurizes a vessel. When that happens, it can be raised right up to about 120 degrees Celsius. And if you hold things at that temperature, even the spores will be killed. So above this temperature, of course, only the hyperthermophiles would survive under normal circumstances. And hyperthermophiles are not of a concern to human beings for any reason. They're at the bottom of the ocean. All right. In addition to temperature, there are pH constraints for the very reasons that I've explained to you already. Many metabolic processes depend upon shuttling ions around. In addition, the structure of enzymes, structure of macromolecules is often dependent upon the formation of these weak bonds, such as hydrogen bonds, which can be easily disturbed if the pH is changed. Remember, pH is a reflection of the proportion of protons in solution. And we change the proportion of protons in solution, we begin to interfere with things which depend on these weak charge bonds. Most bacteria grow in a very narrow pH range, 6.5 to 7.5. An exception to that is that the molds and yeasts tend to be able to grow at slightly lower, but still this is not very, very low. 5 and 6 is not, the 5 is getting a little bit low, but 5 and 6 is not particularly acid solution. But there are some bacteria, especially, which grow at very low pHs. And some of them are actually of interest to us because we have areas of our body which have a very low pH indeed. And for example, in the stomach. There are organisms which we'll hear about again when we talk about diseases of the gut. So we have bacteria which actively grow in our stomachs, which are specially adapted to the low pH of the stomach. And those are referred to as acidophiles. And the pH of the stomach is around about 2, which is extremely acid due to the hydrochloric acid that's present. So those are the acidophiles. They have to be specially adapted. We'll hear about how that is much later on. There are also groups, so these are our acidophiles. They're growing down around about 2, 3. There's the optimum there. And the other group, far more common, pH 6.575, those we refer to as the neutrophiles. They're growing around neutral. And that is by far the majority of the prokaryotes that we are going to talk about. But there are some prokaryotes, especially in that strange group, the archaea, which are adapted to very high pH, 9 and 10, which is caustic to us. And they are specially adapted for that. So these we would refer to anything which grew in such an extreme environment. We refer to as an extremophile. So those hyperthermophiles, those are also extremophiles. They are prokaryotes adapted to these extraordinary conditions which would kill most living things. The next constraint is the osmolarity of the environment, the amount of osmotic pressure which is exerted on an organism by the medium in which it lives, which is a reflection, of course, of the relative amount of solute in the environment relative to the amount of solute inside the cytoplasm. Now, for most organisms, most prokaryotes, live in an environment where the concentration of inside the cytoplasm is either equal to or much higher than its surroundings. So it's either isotonic, which is the case in many environments for bacteria which are living inside a living being, for example. Very often they are living in an isotonic environment where there's as much solute in the environment as inside. But they actually very often prefer for the environment to be slightly hypotonic. That means that the environment has a lower concentration of solutes. Remember back how I described this to you before. That means that the outside has a higher concentration of water. And there is a tendency for the water to diffuse through the membrane into the cytoplasm and begin to swell the cell.
Professor?
Yes, go ahead.
I'm sorry, I have a quick question. So hypertonic is when there is more like solute on the outside or the inside?
You need to specify which solution you're talking about. If I say that the outside is hypotonic to the cytoplasm, then the outside has less solute. That is hypo. If I say that the outside solution is hypertonic, then it means it has more solute than there is in the cytoplasm. I can describe it the other way around. I can say that the cytoplasm is hypertonic to its environment. Okay, so you always must be specifying what is hyper or hypotonic to what. You're always relating two things and what we in fact relating is conditions on one side of the membrane to conditions on the other side. If this is hypertonic out here, there's more solute. If it's hypotonic, there is less solute. So, and then you need to invert your thinking to think about, well, then what's the concentration of water? If this is hypotonic out here, there's a higher concentration of water than there is in here. and the water will tend to go in this direction. It tends to flood into the cytoplasm by osmosis. So what is going to happen now is that that cell is going to begin to swell. Well, this is actually an advantage. It's an advantage in many organisms. It's an advantage in any organism which has a cell wall, because the cell simply swells up against the cell wall until it cannot swell anymore. And then it stops. The net flow of water will stop. And so most prokaryotes and plant cells as well prefer a slightly hypotonic surrounding solution. They prefer to be in a hypotonic environment. And that's by far the majority. Go ahead.
Wait, so what causes osmosis?
I'll explain it again.
Okay.
Okay, osmosis is the diffusion of water. Osmosis is the diffusion of water molecules down the concentration gradient. So now you need to think, what is the concentration? What is a concentration gradient of water? Well, if this is hypotonic, if I've got less solute on this environment than inside here, there is less water inside here. The concentration of water is lower because the concentration of solute is higher. The concentration of water out here is higher because the concentration of solute is lower. Water is going to it's going to behave like any chemical. It's going to diffuse down its concentration gradient. Concentration of water is higher out here and it's lower in here. It's going to diffuse that way. And that's what we refer to as osmosis. Osmosis is simply the diffusion of water molecules, and it's diffusion down a water concentration gradient. So it's diffusing from a high concentration of water to a low concentration of water. Therefore, it's starting to fill the cell. And in the case of prokaryotes, it fills them until they swelled up right up against the cell wall. There's a pressure inside here. And when the pressure inside, which you call the turgor, equals the osmotic pressure, the net flow of water stops. As many water molecules flow in, must flow out. And the organism is perfectly happy. It's on easy street. It doesn't have to do anything more to control the osmotic pressure. The opposite situation is much, much more dangerous for prokaryotes, and it's for plant cells as well. If I take the cell and I drop it into an environment where the outside is hypertonic, in other words, this area outside here has much higher solute concentration. We can think of it as being salt, okay? We take a freshwater bacterium and we drop it into seawater. What is going to happen? Well, in that case, the inside of the cell now has a much higher water concentration. This is hypertonic to this. The water is going to flow that way by osmosis. It's going to flow out of the cell. And the cytoplasm is going to begin to shrink. And it's actually going to pull away from the cell wall. And if the cell can endure this for a while, but if it proceeds and it remains that way for too long, the cell becomes completely disrupted. It's a very, very dangerous situation for these cells. And they, under normal circumstances, they have no way of controlling this. It simply happens passively because the concentration gradient of water now is in this direction. The water flows out and the cytoplasm shrinks. But there are organisms, as we'll hear, there are some organisms, some of them important to us, which are adapted to circumstances like this. They actually have ways of pumping ions and all sorts of things, and are able to actually survive these kind of conditions. And we'll hear about them a bit more. This situation here, when the cytoplasm has shrunk away from the cell wall, is called plasmolysis. And it occurs when the environment is hypertonic. Water leaves the cell, and it is a very dangerous situation for the cell. Growth is severely inhibited under those circumstances. Okay, this is just an explanation of it. There are some organisms which actually require a very high salt concentration. Many marine bacteria, for example, are adapted for the salt concentration of seawater. But there are also some important to us, which grow in salty environments on us, for example, on our skin. Many of the organisms associated with our skin can tolerate a high salt concentration in their environment. So many of those also survive lower, and those we call the facultative halophiles. They tolerate the high osmotic pressure. There are some, especially marine organisms, but also organisms which grow in very, very salty environments, which require a very high salt concentration. And those we call, they're also extremophiles. We call them obligate halophiles. So the facultative halophile is one which can survive and grow at a high salt concentration, but can also grow at lower. Extreme and obligate halophiles require a very high osmotic pressure. Okay, so those are three basic environmental constraints that must be adapted to. All organisms also require a constant input of different elements. And for a start, the most important one of all is carbon, because all life on Earth is carbon-based. All of our macromolecules depend upon a backbone of carbon. But not only that, we also depend upon carbon-based molecules, such as sugars, fats, etc., as a fuel source. So carbon is number one. Carbon is the one thing that we have to have a constant source of. We continually break down carbon in our bodies and release it as carbon dioxide. So our carbon molecules are continually being lost. All organisms require a constant source of carbon. It's this backbone of molecules. It's the source of our energy. And in one group of organisms, which are the autotrophs, the photosynthetic organisms, for example, they require carbon in the form of carbon dioxide. And that is the basis of all of the heterotrophs depend upon the autotrophs, as I explained to you before in the process of photosynthesis. They convert simple carbon dioxide into complex molecules, which then serve as the carbon source and the energy source, but the carbon source for all the heterotrophs as well. The next most important is nitrogen. And nitrogen is a component of many different biomolecules. It's a principal component of proteins, of DNA, and you'll remember of all of the other nucleotides, including ATP, ADP and ATP, and many other molecules as well. So it's easy to understand how we acquire our carbon, or how prokaryotes acquire their carbon. They're actually they're either utilizing carbon dioxide if they're autotrophs, or they're using already manufactured biomolecules as a source of carbon. It's a little bit more difficult to understand where the nitrogen comes from, because nitrogen in the air is in the form of N2, and that is a very, very non-reactive molecule. And ultimately, all nitrogenous compounds, through elaborate cycling through the biosphere, end up being released into the atmosphere, ultimately as N2 again. And because it's so unreactive, it's very difficult to actually grab nitrogen and put it into a biologically useful form. Most things depend upon already created nitrogenous molecules for their source of nitrogen. So, for example, bacteria and fungi, etc., break down protein materials. They decompose protein and things in order to acquire the nitrogen that they need. And some bacteria also are able to use actual gaseous and simple forms of nitrogen, such as inorganic forms of nitrogen, ammonia or nitrate, etc. They can use that. But ultimately, they are going to release the nitrogen. They're going to break it down completely, and it will go back into its forms, which are difficult for biological things to use. We can breathe in nitrogen easily. It's most of the air that we breathe, but it does us no good whatsoever. We can't use nitrogen. We need it to be put into a biologically useful form. Fortunately for us, fortunately for everything, there are groups of bacteria which can take atmospheric nitrogen, N2, and they can actually put it into a biologically useful form. And in fact, we all depend upon them for a continual supply of nitrogen being fed into the biosphere. So nitrogen is a very interesting element. We aren't going to discuss it in huge detail, but its cycling through the biosphere is very, very complex. And it is almost entirely dependent upon prokaryotes to ensure that nitrogen is continuously available to all other living things. A couple of others, we require sulfur, especially it's because it is used, it's a component of some of the amino acids. It's also a component of some vitamins and a nitrogen, of hydrogen carriers, reducing power. So it actually participates in a number of different biomolecules and different metabolic processes. And again, we require a source that most heterotrophs acquire their sulfur from their food. And there are some very interesting prokaryotes which are actually able to break down protein, for example, deliberately as a source of sulfur. Others can use inorganic sources, and they use things like sulfate ions. They use hydrogen sulfide as sources of sulfur. One of the really important functions of sulfur that you should remember is this involvement in amino acids. The reason it's so important is that certain amino acids with sulfur groups can actually bond with one another. They form what is called a disulfide bridge. Two sulfurs join together, and it's a very strong covalent bond. And in proteins, that is used to stabilize the three-dimensional structure of the protein. So that's why that is one of the reasons that sulfur is so important. The next one is phosphorus. Phosphorus is found especially in the nucleotides. And you'll remember the structure of the nucleotides. They're a sugar, a ribose sugar of some sort, or a deoxyribose sugar with a nitrogenous group. There's where a lot of nitrogen is used. But then they also have a phosphate group. And the structure of the nucleic acid is built from the phosphates of one nucleotide joining onto the sugars of another. So these phosphate groups are extremely important in structuring of the nucleic acids, DNA, RNA. But those phosphate groups are critically important to all living things because of those nucleotides which are used to transfer energy. And that we talked about extensively. This was the nucleotide ATP, adenosine triphosphate. And that last triphosphate, it has 3 phosphate groups. The energy associated with that last phosphate bond is the most important of all biological sources of energy for driving metabolic processes. Okay, in addition, many things, prokaryotes, I should tell you, are very, very good at manufacturing almost everything they need. But there are some which require external sources of organic compounds, and those are called essentials. So they are essential organic compounds. They're things which must be acquired from the environment. There are limitations on the synthetic ability of most living things. There are certain things they cannot make themselves. And when we talk about prokaryotes, us as well, but especially in prokaryotes, there are certain vitamins and amino acids, some purines, some pyrimidines, which they need from outside. They need to be supplied, and those are usually supplied in their normal environment. They're supplied from food sources. When we culture prokaryotes, however, we need to remember this, because sometimes we have to supply them, especially if they're specialized bacteria. We may need to supply things to them that they are unable to synthesize, and those we refer to as being essential nutrients. Another set of essential nutrients are the trace elements. These are inorganic elements required, as the name says, trace elements. They're usually only needed in very, very small quantities. There's one good example for you, and that is iron. We require iron. Why? Because it's part of hemoglobin. It's part of the enzyme hemoglobin. It functions together with hemoglobin. And it's only needed in small amounts, but essential amounts. And they are functioning as enzyme cofactors. And there are a number of them. Iron is the one I mentioned, but copper, molybdenum, and zinc are all trace elements that most living things require from their environment as well. Okay, so here's a really important environmental constraint, and that is the presence and absence of oxygen in the environment. So I should explain to you at this point. I may have done this before, but it bears repeating. The origins of life were in an anaerobic environment, in an environment which was completely devoid of free oxygen. By free oxygen, I mean either a single atom, O, or one of the other combinations. O2 is the commonest, but sometimes other species of oxygen can also occur. But O2 is the commonest species of oxygen present in our atmosphere and in our environment. But that is not the way it always was. When the planet was first formed, all of the oxygen present on the planet was bound up in with metal ions, such as iron. It was all bound up as iron oxide and carbon dioxide and water, and it was bound into compounds on the planet. There was no free oxygen. So life itself evolved under conditions where there was no free oxygen. And sometime later, about 500 million years to 750 million years later, a group of prokaryotes evolved the system of photosynthesis. They evolved a system where they could take carbon dioxide and water, put them together to form sugars. That's great, because they were using the sunlight. They were binding carbon dioxide, hydrogen together with the energy from the sun. That's the process of photosynthesis. But what is the byproduct of photosynthesis? The waste product of photosynthesis is free oxygen. And this was the very first great extinction event on the planet. This was one of the first great crises for life on the planet. The system of photosynthesis worked extremely well for those organisms which could perform it, but there was a tremendous problem, and that was that the oxygen levels began to increase. And oxygen is a very dangerous molecule because it burns through, it will spontaneously burn through biomolecules. It is extremely damaging to biosystems under normal circumstances. And in actual fact, we believe that the system of aerobic respiration, of taking oxygen and burning fuels with it to make carbon dioxide and water, was a response to the dangers of oxygen. It was a way to survive the presence of oxygen. So the story of oxygen and life is extremely complicated, and it's a very interesting one. And there were many evolutionary steps involved in generating oxygen. Almost all the oxygen we breathe is generated by living things, by the processes of photosynthesis, of autotrophy. But in order to survive in an atmosphere with oxygen, then a living thing must have strategies for dealing with the oxygen. Most have resorted to the processes of aerobic respiration, which is exactly what we do. Of course, we benefit enormously because it evolves into the system that produces masses of ATP for us. So that was great, and also deals with the oxygen. But there are other organisms which never evolved those systems, and they had to adopt other strategies. So when we look at all, especially at the prokaryotes, what we see is a range. Most are organisms which are tolerant of oxygen. And so they have aerobic respiration, and we say they are aerobic. Some of them positively require the presence of oxygen. They cannot survive in the absence of oxygen. Those we call the obligate aerobes. Don't worry about if you're writing it down, that's fine, but we'll hear these terms again as I go through this chart. So the ones who absolutely require oxygen and cannot survive in its absence are called the obligate aerobes. There are many prokaryotes which preserve their early metabolic processes, which enable them to survive in the absence of oxygen or in its presence. So if they can survive in both the presence of oxygen and in its absence, we say they are facultative anaerobes. So just think back to our sections on metabolism. We talked about aerobic respiration, right? Where we burn fuels with oxygen to produce energy. But what was the very start of those processes? The very start was glycolysis. Glycolysis is like a fossil metabolic pathway. It's a reflection of how organisms functioned in the total absence of oxygen. All of those early steps, no oxygen was used at all. And there are many, many prokaryotes which preserve the ability to resort to just glycolysis to survive if oxygen is absent. If they can do both, we call them facultative anaerobes. But there are those organisms, those prokaryotes, which are unable to survive in the presence of oxygen. They have no way of dealing with the damaging effects of oxygen. So they must exist in the complete absence of oxygen. And those we call the obligate anaerobes. And there are a couple of groups which are really, really important, especially in human health, which we will hear about as we go along. So with that lengthy explanation, preamble, I'd say, let's now do an experiment. You may, in fact, end up doing things like this in your lab. What we're going to do is we're going to set up an experiment to look at the impact of oxygen on different things, different microorganisms. We're going to take a test tube. We're going to put in it a medium. It's usually a medium with a little bit of agar in it, so it's almost a little bit jelly-like. So that that is just so that oxygen doesn't diffuse very, very easily through it. We also often will put into it something which will largely sop up all the oxygen. So that what we have here is a medium, which when we first make it is anaerobic. It has no oxygen in it, except at the very, very top, where oxygen in the air is present. We're going to leave that. It will actually establish a gradient of oxygen. There will be some oxygen up at the top, dissolved in. There'll be very little oxygen in the middle, and there'll probably be no oxygen down here at the bottom. That's what the tube ends up looking like. And then we're going to put into that, we're going to inoculate it with different bacteria. And we're going to see where in that medium they grow. So here's our first tube. We inoculate it with bacteria, we leave it, we incubate it for 2 days or whatever, and we come back and look and see where is it growing? Where is that organism growing? And you see it's only growing here at the very, very top there. And what is our diagnosis? It's only growing where oxygen is relatively abundant, right at the very surface of the medium. That is typical for an aerobe. Its growth is aerobic, and it requires oxygen. We refer to that as an obligate aerobe. And air, it's spelled A-E, but you can think of it as, it should remind you, aerobe, something that likes air, something that likes oxygen. So this organism has no ability to resort to glycolysis only. They have glycolysis because glycolysis is the start of that whole process of constructing abundant ATP from it during aerobic respiration. They all have some pathway like that, but they are unable to simply use glycolysis. It's not enough for them to survive or grow. So they only grow where they can perform aerobic respiration. In addition to that, these organisms possess other enzymes. And these are enzymes to deal with very, very dangerous species of oxygen, which are produced especially during the process of respiration, but which do also occur naturally. And these are things like O3 and hydrogen peroxide and things like this that are very dangerous. They burn through biomolecules. And these organisms possess enzymes which will deal with those as well. The only ones you need to remember, the enzymes, catalase and superoxide O3 dismutase. So superoxide is O3, and superoxide dismutase converts that into a product that can be dealt with. Okay, so there's our first one. It's relatively easy. Here's our next one. This is another organism. They're inoculated into the tube under exactly the same circumstances, and we see where it grows. Well, you can see here it's got heavy growth at the very top, but it's grown throughout. But the way in which it's grown is interesting. It's grown very heavily here. Why? Well, because aerobic respiration is extremely efficient, produces masses of ATP, right? When we move this organism into an anaerobic environment, aerobic respiration stops. And the organism growing in the rest of the tube has to resort to glycolysis. And that glycolysis is enough to keep the organism going, it's enough to keep it growing and reproducing, but only slowly, because it only produces a small amount of ATP. And so this is typical. These facultative anaerobes. There's the name there. A facultative anaerobe usually grows much better in the presence of oxygen, but will continue to grow in the absence of oxygen. They can utilize aerobic respiration up here and glycolysis down here. They also have enzymes which will neutralize those dangerous species of oxygen that they might encounter, especially up here. Okay, so the next group, here's another kind of bacterium, and I'll tell you the name for this one. This is Clostridium, okay? That's the typical one to use. We inoculate it, and the only place it grows is all the way down here at the very, very bottom. That's the only place it will grow, in the complete absence of oxygen. In fact, oxygen is toxic to it. And this is an obligate anaerobe. And the obligate anaerobes are those which depend entirely upon the processes like glycolysis to manufacture their ATP. They depend upon completely anaerobic processes in order to generate ATP in the cell. They also lack any of the enzymes which would deal with dangerous species of oxygen. And these organisms are interesting because they are like fossils. This is what the first prokaryotes must have looked like. This is what the first prokaryotes must have functioned like, because there was no oxygen present. So here, this next group is a group you seldom see, actually. You don't deal with them very often. They're called the aerotolerant anaerobes. It doesn't make much difference to them whether there's oxygen present or not. Their growth is about the same. They tolerate oxygen, but they largely depend on pathways like glycolysis to survive. But they don't have, they gain no advantage from the presence of oxygen. So they are using something like glycolysis or the Entner-Duderoff pathway or something like that to generate their ATP. But they do possess some enzymes which will help them deal with dangerous oxygen species. So oxygen is not actively damaging to them. And then the last group are interesting for us because we do see them in some pathogenic bacteria. And these are called the microaerophiles. Here again, we have our tube, we inoculate it. And here, they grow only in a little narrow band. And it looks like they can tolerate a small amount of oxygen. In fact, they require a small amount of oxygen. They do perform aerobic respiration. But the thing is that they lack the sufficient enzymes to deal with dangerous species of oxygen. So they are wiped out up here. And they cannot resort to glycolysis only. So they cannot grow down here. They get constrained to a low oxygen atmosphere. Well, just think about a pathogenic bacterium circulating in the body. Many, many places in the body, a pathogen is going to be existing in an atmosphere of very low oxygen. And for this reason, there are those microaerophiles which which we would find as pathogens. They are perfectly able to survive under low oxygen concentrations, but not under very high or very, very low or absence of oxygen. Okay, so be sure you will understand from the way in which I have gone through this that this is an important section. Okay, know each of these and know what it is. What is these growth patterns? Why did they arise like this? And what is it about the physiology of these organisms that constrains them to these particular oxygen environments? I will just tell you that the vast majority of things that you deal with in lab, for instance, are either obligate aerobes or facultative anaerobes. Some of these ones here are a little bit more difficult to deal with in the lab. They may be really important in health terms and everything, but they're somewhat more difficult to deal with under lab conditions. All right. So let me just divert a little bit and explain that I've talked so far about these organisms as if they're just by themselves in the environment, entirely dependent on their environmental conditions for their rates of growth and everything. But let's assume that they have enough, they have the correct oxygen environment, they have the correct pH, enough food, etc., etc., etc. There's one thing that we haven't talked about, and that is that they also have to interact with one another. And we're going to have bacteria of the same species must interact with one another, and bacteria of different species must interact with one another as well. Often bacterial environments are extremely rich, extremely species rich. And the way in which they interact with one another is very complex and often very interesting. Unfortunately, we don't have a lot of time to discuss these aspects of microbial ecology. But there are a few things that we need to mention because they are so important in terms of health sciences, especially. We need to accept that when we look at bacteria growing on or in another organism, there are two situations. The first that we talk about often, the first thing that springs to mind, oh yes, bacteria are pathogenic, they cause disease. Well, that is prokaryote acting as a parasite, causing harm to its host in order that it can benefit. But many, many bacteria that we are involved with are not pathogens at all. They are part of a normal bacterial flora that inhabits us and lives with us, and is actually of enormous benefit to us. And these don't exist usually as a single species. They exist as a mixture of species that interact with one another, and they form a true microbial community. So the first thing to explain to you is that bacteria can actually communicate with one another. And we see this when we grow bacteria. When we grow bacteria in a medium, for example, we'll see later on in detail how this happens, but we'll see that when we first put them into the medium, their growth is extremely slow. It's not just that there's only a few of them, and so we can't measure many. If we look at the actual growth of the cells, it's very slow. They do grow, but they divide slowly and they grow slowly. But they slowly increase in numbers. As they increase in numbers, we see something really strange happening, and that is their growth rate begins to increase. And the reason for this is that the bacteria are actually secreting substances which inform the other bacteria that they are present and inform the other bacteria about their metabolic status. We call this quorum sensing. A quorum is a requisite number of people in politics. The quorum of a committee. You have to have a requisite number of people present for a vote to be valid, right? That's a quorum. In bacterial communities, a quorum is sufficient bacteria present that they can signal to one another that conditions are exactly right and growth can proceed unimpeded. And this they do, they secrete inducer chemicals. These are signaling chemicals. And those signaling chemicals do two things. They first of all inform other bacteria about the environmental status. But second of all, they appear also to attract bacteria. And they may attract not only their own species, they may attract other species of bacteria, and they all begin to interact with one another. And they interact in positive ways. These communities overall are much healthier than groups of just individual species. These communities of bacteria confer advantage on one another. And very often what they're doing is they will shelter the bacteria from harmful environmental factors, especially things like predators, things that come and eat them, and also things like our immune system, things like that. So here's when we look at these communities, they especially form on surfaces. And those surfaces can be artificial surfaces, but they will form on surfaces such as our respiratory surface, our gut surface as well. But they don't just form a thick, impenetrable layer. Instead, they begin to form a structured community, which allows percolation of the medium, for example, through the community. They can start to bind to, first of all, to the surface that they're attracted to, but they also begin to bind to one another. And that is through those external structures that we saw before, things like the slime layers, the capsules, all of the fimbriae, all of these things begin to bind the bacteria together to form a biofilm, a thick layer of the complex community of bacteria. And just we won't discuss it in major detail, but just remember that these bacteria may be doing different things, and they may have different requirements. So, for example, the bacteria on the outside may be obligate aerobes. The bacteria further in may be facultative anaerobes. The ones right in the middle, right there, right in the center, may be obligate anaerobes. And they are maybe protected there by the fact that the outer bacteria, for example, are using all the oxygen before it percolates through the biofilm. That simple principle applies in many things. We may have bacteria which are producing species of nitrogen, which the others require. They may be.
Professor?
Yes, go ahead. Go ahead.
Sorry, I have a quick question. So the biofilms, is it the layer outside of the bacteria, or is it the thing that the bacteria is attaching to?
No, this whole thing here, the whole structure formed by the bacteria all sticking to one another and to the surface. That's the biofilm.
So it's everything?
Yes, this whole thing here, that whole thing there is a biofilm. So it's not, and just remember, it's not a solid layer. It's structured like this, but it's all communicating with one another.
Is it like a liquid substance?
Think more gel. Think it's more like jelly-like substance. This is surrounding here the individual cells, the individual bacterial cells. All of this stuff surrounding them is capsule material. It's all polysaccharide, like mucus, like thick mucus, and slimes, which are creating the film itself. And all of the bacteria are embedded in it, except for where they stick here. You may have bacteria that are actually adhering to the surface here.
And this is any surface?
Virtually any surface, yes, that's surrounded by a medium will begin to build up a biofilm. So, for example, we have a biofilm through our whole upper respiratory tract. We have a biofilm through our respiratory tract almost from the bronchi all the way up. We have a biofilm throughout our entire gut, down through the esophagus, minor in the stomach, because not much survives there, but then in the small intestine, the large intestine, the whole thing is layered with a biofilm. But that's a healthy, under normal circumstances, that's a really healthy situation. In the gut, the respiratory tract, this is what we call the microbiota. And it's the subject of a lot of research, because it turns out that It's extremely intimately involved in our health. To have a healthy microbiota protects you from many, many disease situations. So these become very, very important. This is an important interaction between prokaryotes and their hosts. We'll hear a bit more about it later on, especially when we talk about gut bacteria. Okay, so it doesn't only, these will not only form in living systems as we have for ourselves, they will also form on virtually any surface. You get these very complex bacterial communities building up. Now, these biofilms are important to us for various reasons. They're important to us because we have healthy biofilms, which are part of our structuring. But they become important under health systems because they will very easily form on artificial surfaces as well. And they may be formed from bacteria which are highly undesirable. And when they do form, they can become extremely difficult to deal with because they can be very, very resistant to disinfection, to microbicides, and all of those sorts of things. When you look at hospital infections, at the types of infections which people acquire just because they were in hospital, those we call the nosocomial infections. 70% of nosocomial infections are associated nowadays with biofilms forming on medical equipment. This is extraordinary, but it's true because we actually increasingly insert into human beings artificial structures. We put in heart valves. We put in stents. We put in artificial joints, et cetera, et cetera. So when somebody is in a hospital for a long time, they may have a catheter inserted. They may be having respiratory assistance, as many of the severely ill COVID patients do, by having a tube inserted into their bronchus, and on and on and on. All of those artificial structures are liable to become populated with a biofilm of some sort. And very often in those situations, what builds up on the catheter or whatever are undesirable bacteria, which can actually cause disease. The one which is that most of you need to be careful of, first of all, if you use contact lenses, this is why you have to be so observant for hygiene on contact lenses, because contact lenses very quickly build up a biofilm, and it may be of undesirable things. The other one, which affects virtually all of us, the deleterious biofilm that affects all of us, is the biofilm that builds up on our teeth. And if our diet is incorrect, if we, for example, eat too many sugars, and that sort of thing, we change the structure of the biofilm on our teeth, and we start to favor organisms which produce acid. And that is what decays our teeth. So dental caries is due to the activity of the biofilm as well. I think here's a picture of one. This is a picture of this is in this case a single organism called Serratia, and this is it's formed on a piece of plastic. Here's the plastic, but here's this. You see all these strands. This is all capsule material and fimbriae and things which are tying the bacteria on the bottom to the surface, and then other bacteria are growing together with them to form this thick film. So this is a good example, because Serratia is not it occurs naturally. It occurs naturally on our skin and everything, but it's particularly favored by this kind of artificial situation. And if this is a catheter, which is quite possibly is a urethral catheter, which is being inserted to empty somebody's bladder, and it's resident, it's being left in there for a long time, Serratia can cause an infection of the bladder. It causes cystitis, and that cystitis can be extremely serious because it can also ascend to the kidneys. So it becomes very, very important that things like catheters be regularly checked and that their hygiene be really carefully monitored, because they can produce dangerous results. And as we saw, a very high proportion, 70% nosocomial infections arise because of bacteria associated with surfaces of these things. Okay, let's just leave it there for today, and I'll start on this slide the next time we meet. And then please also don't forget that you have your second test coming up next Wednesday, again online. Now, look at your calendar. It says they're midterm, but this is a normal test. Okay, this is a normal class test. It covers material that you have not been examined on yet. So it's up till the end of our class today. I won't test you on stuff done on Monday, but up to the end of today's class, up to this slide that has not been examined, is fair game for the second test. All right. Are there any other questions?
You said up to this class is the exam?
Yes, up till this slide, not including this slide, up to this slide.
Yeah, okay.
Okay. Thank you so much, everybody. I'm always very gratified that so many people do log on for this, for the live class. And I hope, oh, sorry, I have a question. What is a microbicide? A microbicide is anything which will kill a microbe, basically. So usually when we talk about microbicides, we mean things that are applied to the bacteria externally, not something like an antibiotic, but disinfectants, all of those sort of things. Those are microbicides. Microbicides, basically anything that will kill bacteria. Okay, and I'll just type that in. Okay, everyone, thank you very much.
Thank you.
Thank you, Professor.
Bye. Have a nice day.
Thank you.
You too.